On a September morning in 1928, a Scottish bacteriologist returned to his laboratory at St Mary's Hospital in London after a summer holiday and found something growing on a petri dish that he had left on his bench before leaving. This was not unusual. Contamination of bacterial cultures was a persistent and irritating feature of laboratory work in the 1920s. Stray mold spores drifting through the air and landing on agar plates were common enough that most researchers simply discarded the contaminated dishes and started again. The contamination was a nuisance, not a discovery. But this researcher did not discard the dish. He looked at it more carefully. And what he saw, the specific pattern of clear space around the mold colony where the bacterial growth had been inhibited, was something that most other scientists would have dismissed as a routine contamination artifact and something that his specific mind, prepared by 20 years of medical research and a specific intellectual orientation toward the unexpected, recognized as potentially extraordinary. His name was Alexander Fleming. The mold was Penicillium notatum. The bacteria it was killing were Staphylococcus aureus, the bacterium responsible for blood poisoning, wound infections, pneumonia, and a range of other conditions that were killing millions of people every year at a time when medicine had no reliable way to fight bacterial infection. And the substance the mold was producing, the substance that was creating the clear halo of death around the mold colony on Fleming's petri dish, would eventually be named penicillin. It would not be available as a medicine for another 13 years. It would not be mass-produced until a world war made the urgency unavoidable. It would not be universally acknowledged as the discovery that it was until two other scientists, Howard Florey and Ernst Boris Chain, did the chemical isolation and the clinical testing that transformed Fleming's laboratory observation into an actual drug. And the specific complicated story of who deserved how much credit, the discoverer who found the mold, the biochemist who purified the drug, the pharmacologist who tested it, would generate controversy and bitterness, and the specific human drama of a Nobel Prize shared three ways in 1945 that not all three recipients felt was equitably distributed. But the story begins before Fleming. It begins, as all great scientific stories begin, in the accumulated context of the science that preceded the discovery, in the specific intellectual landscape of bacteriology and immunology in the early 20th century that prepared the ground for what Fleming found on his contaminated plate. The germ theory of disease, the specific revolutionary understanding that infectious diseases were caused by specific microorganisms, not by miasma or constitutional weakness or divine punishment, had been established by Louis Pasteur and Robert Koch in the second half of the 19th century. Pasteur's experiments with fermentation in the 1850s and 1860s, his development of the theory of microorganisms as agents of specific diseases, and his pioneering work on vaccination had established the fundamental conceptual framework. Koch's development of specific laboratory techniques for isolating and identifying pathogenic bacteria, including the specific methods of growing bacteria in pure culture on solid media that gave the subsequent generation of bacteriologists their most important experimental tools, had provided the methodological foundation. By the early 20th century, bacteriologists could identify the specific bacteria responsible for most major infectious diseases, could grow them in pure culture, could study their behavior under laboratory conditions. What they could not do, in most cases, was kill them inside a living patient without simultaneously killing the patient. The search for substances that could kill bacteria selectively, that were toxic to microorganisms without being equally toxic to the human tissue surrounding them, was the central challenge of early 20th century medicine. Paul Ehrlich, the German physician and chemist whose concept of the magic bullet, the specific chemical substance that would bind selectively to a pathogen and kill it without harming the host, was the guiding metaphor of the entire field, had achieved the first success with salvarsan in 1909. A compound of arsenic that was effective against the syphilis bacterium and that represented the first demonstration that systematic chemical screening could find compounds with selective antimicrobial activity. But salvarsan was toxic. It required careful dosing and careful administration. It worked for syphilis but not for the far more common bacterial infections, the streptococcal throat infections, the staphylococcal wound infections, the pneumococcal pneumonias, that were killing the majority of people who died from bacterial disease. The magic bullet for common bacterial infections remained unfound. Alexander Fleming was born on August 6, 1881, in Lockfield, Ayrshire, a farm in the rural lowlands of Scotland, the seventh of 8 children of Hugh Fleming and his second wife Grace Morton. His early childhood was the specific rural Scottish childhood of the late Victorian era, working the farm, walking the moorland, developing the specific practical relationship with the natural world that farming communities build across generations. His father died when Fleming was 7, and his older brother Tom, who had trained as a physician and was establishing a medical practice in London, became the dominant educational influence in his life. He came to London at 13 to attend the Polytechnic in Regent Street, began working as a shipping clerk, and in 1901, prompted by Tom's encouragement and the legacy of a modest inheritance, entered St. Mary's Hospital Medical School to study medicine. He qualified as a doctor in 1906 and joined the research department of St. Mary's under Almroth Wright, the distinguished immunologist and vaccine developer whose theories of immune response shaped the intellectual environment in which Fleming's early research was conducted. Wright's laboratory was a specific intellectual world that shaped Fleming in ways that both enabled and limited his subsequent career. Wright was a brilliant and dominating figure who believed passionately in the primacy of the body's own immune defenses, that the proper goal of medicine was to enhance the patient's natural immunity rather than to attack bacteria directly with chemical substances. This specific intellectual orientation, which inclined Wright toward vaccines and antisera and away from the direct antibacterial chemistry that Ehrlich was pursuing, was the philosophical context in which Fleming worked for his entire career at St. Mary's. Fleming's most important pre-penicillin contribution to science was the discovery of lysozyme, an enzyme naturally present in human tears, saliva, and other secretions that had antibacterial activity. Fleming discovered lysozyme in 1921 when he noticed that a drop of his own nasal mucus, introduced into a bacterial culture when he had a cold, inhibited bacterial growth. He spent years studying lysozyme's properties, establishing its presence in a wide range of human tissues, demonstrating its antibacterial activity, speculating about its role in the body's natural defenses. The lysozyme work was important as science and as preparation. It trained Fleming's eye to see and value antibacterial activity in unexpected sources. It established in his mind the specific category of naturally occurring antibacterial substances, substances produced by living organisms as part of their own biological activity, that penicillin would eventually exemplify at a vastly more powerful level. And it gave him the specific experimental habits and the specific intellectual orientation toward naturally produced antibacterial compounds that made him, of all the bacteriologists in the world, the person most likely to recognize the significance of a mold-killing bacteria on a contaminated plate. The summer of 1928 and the contaminated plate. The specific details of how the contamination occurred, which remain somewhat uncertain despite decades of historical investigation, involve the specific geography of St. Mary's Hospital and the specific meteorology of London in the summer of 1928. The Penicillium mold that contaminated Fleming's plate was not a common environmental contaminant. It was a relatively unusual strain whose specific antibacterial properties were far superior to those of most Penicillium species. The specific combination of an unusual mold spore drifting into Fleming's laboratory from a neighboring lab where mycologist C.J. Lartuche was growing mold cultures, landing on a plate of Staphylococcus culture, and being found by the one researcher in the hospital whose specific intellectual preparation made him capable of recognizing its significance, is the specific accumulation of coincidences that the history of scientific discovery occasionally produces and that Fleming himself would later describe with the characteristically Scottish understatement of a man who had thought carefully about the nature of luck. He did not shout Eureka. He called his colleague Stuart Craddock to look at the plate and said something to the effect of "Take a look at this." Then he began systematic investigation. He identified the mold as Penicillium. He demonstrated that the substance the mold was producing, which he named penicillin in his 1929 paper, was not the mold itself but a soluble compound that the mold released into the surrounding medium, and that this compound inhibited the growth of a remarkable range of gram-positive bacteria while leaving gram-negative bacteria relatively unaffected. He tested penicillin's toxicity on animals and found it remarkably non-toxic compared to other antibacterial substances known at the time. He speculated in his 1929 paper that penicillin might have clinical applications, might be useful for treating infected wounds, might be used to selectively grow certain bacteria in mixed cultures, might represent a new class of antibacterial agent. And then, largely, he stopped. The specific question of why Fleming did not pursue the clinical development of penicillin in the decade between his 1929 paper and the Oxford work of Florey and Chain that began in 1939 is one of the most analysed and most debated questions in the history of medicine. The honest answer appears to involve several factors simultaneously. Fleming was a bacteriologist, not a chemist. The specific challenge of extracting and purifying penicillin from the mold's culture medium was a chemical problem that he did not have the training or the equipment to address. The penicillin in the culture fluid was unstable. It degraded rapidly when attempts were made to concentrate it, making it difficult to work with. Fleming's attempts to enlist the help of chemists at St. Mary's to solve the purification problem were unsuccessful. And the specific intellectual environment of Wright's laboratory, with its emphasis on immune stimulation rather than direct antibacterial attack, may not have provided the strongest incentive to pursue what was essentially a chemotherapy approach. The story passed to Oxford. Howard Florey was an Australian-born pharmacologist who had become Professor of Pathology at the Sir William Dunn School of Pathology at Oxford University in 1935. Ernst Boris Chain was a German-Jewish biochemist who had fled Nazi Germany in 1933 and joined Florey's department at Oxford. The specific combination of Florey's organizational and experimental abilities and Chain's exceptional biochemical skill, and the specific funding they received from the Rockefeller Foundation in the United States, which was supporting promising biomedical research in Britain, created the conditions for the systematic chemical and pharmacological investigation of penicillin that Fleming's laboratory had not been able to conduct. Florey and Chain began working on penicillin in 1939. Chain had encountered Fleming's 1929 paper during a systematic literature review of naturally occurring antibacterial substances, and the specific properties Fleming had described, the remarkable antibacterial activity and the apparent low toxicity, made it an obvious candidate for systematic investigation. The chemical challenge was formidable. Penicillin was present in the mold culture medium in tiny concentrations. It was chemically unstable at room temperature and rapidly degraded under many of the conditions used in standard chemical extraction. Producing even small quantities of purified penicillin required the processing of large volumes of culture medium under carefully controlled conditions. Chain and his colleague Edward Abraham developed the freeze-drying technique that preserved the purified penicillin in stable form and produced, by the spring of 1940, enough purified material for the first animal experiments. The mouse protection experiment of May 25, 1940, in which 8 mice were infected with a lethal dose of streptococcal bacteria and 4 were treated with penicillin while 4 received no treatment, produced the most dramatic experimental result in the history of antibiotic research. The 4 untreated mice died within 17 hours. The 4 treated with penicillin survived. Florey reportedly said, looking at the surviving mice, "It looks like a miracle." It was not a miracle. It was biochemistry. But the specific visual drama of 4 dead mice and 4 living ones, separated only by whether they had received the substance that a Scottish bacteriologist had noticed on a contaminated plate 12 years earlier, was as close to a miracle as empirical science gets. The clinical testing required much larger quantities of penicillin than Florey's laboratory could produce. The Oxford team converted their entire laboratory building into a penicillin production facility, growing the penicillium mold in every available container, including hospital bedpans, in conditions that the word industrial would glorify but that produced enough material for the first clinical trials. The first patient to receive clinical penicillin was Albert Alexander, a 43-year-old Oxford policeman who was dying of a catastrophic staphylococcal and streptococcal infection that had begun as a scratch from a rosebush. He improved dramatically with the first doses of penicillin, and relapsed and died when the supply ran out before the infection was fully controlled. The specific tragedy of the first penicillin patient dying not because the drug did not work but because there was not enough of it was the clearest possible demonstration of the urgency of the production problem. The Second World War provided the urgency. The specific military demand for a drug that could prevent wound infections from killing soldiers, the specific reality that in previous wars more soldiers had died from infected wounds than from the wounds themselves, created the political and industrial will to solve the production problem on a scale that peacetime medicine had not been able to generate. Florey traveled to the United States in 1941 and enlisted the help of the American pharmaceutical industry and the U.S. Department of Agriculture's Northern Regional Research Laboratory in Peoria, Illinois. The Peoria laboratory made the discovery that transformed penicillin from a laboratory curiosity into a mass-producible medicine. The discovery that adding corn steep liquor to the growth medium dramatically increased the mold's penicillin yield, combined with the isolation of a new Penicillium strain from a moldy cantaloupe found at a Peoria fruit market that produced dramatically higher penicillin yields than Fleming's original strain. The combination of the improved medium, the better-yielding strain, and the development of deep fermentation techniques, growing the mold in large submerged tanks rather than in surface cultures, made industrial-scale penicillin production possible. By D-Day in June 1944, when the Allied forces landed on the beaches of Normandy, sufficient penicillin was available to treat every Allied soldier who needed it. The specific comparison between the wound infection mortality rates of the First World War and the Second is the most direct measure of what penicillin accomplished. In the First World War, approximately 18% of soldiers with serious wounds died of infection. In the Second World War, the rate was less than 1%. Fleming, Florey, and Chain shared the Nobel Prize in Physiology or Medicine in 1945. The Nobel Committee citation described the discovery and development of penicillin as one of the greatest advances in medicine in the history of the world. They were right. The subsequent development of the antibiotic era, the systematic screening of natural and synthetic compounds for antibacterial activity that penicillin's success inspired, producing streptomycin and tetracycline and erythromycin and the dozens of other antibiotic classes that have transformed the treatment of infectious disease across the second half of the 20th century, has saved more human lives than any other medical advance in history. The specific number is difficult to calculate. The lives saved by penicillin alone, not counting the subsequent antibiotics whose development it inspired, has been estimated at 200 million. 200 million people who would have died of bacterial infections that were, before 1943, essentially untreatable. 200 million lives from a contaminated petri dish that a Scottish bacteriologist found on his laboratory bench after a summer holiday and almost discarded. He did not discard it. He looked more carefully. And the world is 200 million lives larger for the specific quality of his looking. The mold is still with us. The bacteria, unfortunately, are evolving. The specific crisis of antibiotic resistance, the evolutionary response of bacterial populations to the intense selective pressure of antibiotic use, producing strains of bacteria resistant to the drugs that penicillin's discovery inspired, is the specific challenge that the 21st century inherits from the 20th century's greatest medical triumph. The bacteria are doing what bacteria do. They are evolving. They have been evolving for 3.5 billion years. They are very good at it. The contaminated plate that Fleming found in September 1928 bought humanity 100 years of relative safety from bacterial infection. The next 100 years will require something equally unexpected from an equally unpredictable source. Somewhere, in some laboratory, on some petri dish, in some unexpected place where a prepared mind is paying attention, the next discovery is waiting. History suggests that it will not announce itself. It will simply be there, growing quietly on a forgotten plate, waiting for the specific quality of attention that changes everything.